So a wide variety of molecules, as I've shown you. This is a table from one of your textbooks that highlights some of the synthesis processes in the center column, and then some of the major effects for these different categories of hormone molecules. Um, I don't expect you to really know these, but it kind of all together maybe gives you a fairly complete picture of what kinds of responses a plant overall and different parts, the major organs of those plant, what kind of responses they would have for any variety of scenarios. So the plant has the ability for given types of cells to stimulate growth, to pause growth. Some of these actually cause cells to die if they get the right kind of signal. And so this is a nice organismal way to manage all the different parts of the plant by producing and sending out these different signals. So there's a lot of control over what the plant is doing within its plant body using hormones. So another illustration here, this is from that same paper that I highlighted before, just showing some of the major classes of hormones, where they're found. Just, I don't know, take home from this is there are lots of kinds of hormones and they're found in lots of parts of the plant, right? No big surprises there. So just thinking about the kinds of signals that plants need to undergo. So during development, what point are you investing more in growing, producing leaves, producing flowers, producing seeds and fruits, ripening your fruits, dropping fruits, and so on. All these would be developmental signals that might need to be queued up with timing or resource availability or something like that. Environment is also giving lots of signals. So we'll see a little bit more in the next topic when we talk about response to the environment, different ways that plants can also respond to environmental signals. But this is also an area where plants would maybe need to communicate to the whole plant or different parts of the plant that there's some kind of damage or opportunity in the environment that can be achieved by hormones. So plants don't really have this nervous system. They don't have a brain. They don't have any kind of central processing center. So instead, it's different parts of the plant as they are able producing these signals to distribute to the other parts of the plant that might respond to the signals. So what can be your responses? Again, I kind of paint plants as rather simple organisms in this class. Not much that is available to them. So you're an individual plant cell, you have the option of growing, stopping growing, dying, um, perhaps producing other molecules, or firming up, differentiating into different cell types and so on. But not a whole lot of options here. So cells along the way in this example are getting the signals for developing from early seedling to later seedling stage. Some hormones involved in that process. And I'd say as far as hormones, we're going to see them by name, see the big categories of molecules. This is, I would say, probably the biggest category of plant molecules, the one that most people would be familiar with, the one that has one of the more obvious effects. And it's a category of molecules, there's several of them, and they're all known as the auxins. So there's one that's a little more commonly known as auxin, but other ones that are kind of familiar, similar molecules to that first one. Auxin actually gets its name from Latin and the same origin of the word augment, right? So to make something bigger, augment it. Auxin is this hormone involved in growing. And that was how it got its name, and that's one of its major functions, is encouraging plants to grow. So growth involving cell division and increase in size. And a nice illustration here of two of these model organism Arabidopsis plants, one that's the wild type version and one that is unable to respond to the signal of auxin. So same environment, same lights and resource availabilities, and this little plant just isn't able to get the signal that it can grow bigger. So it's not growing bigger because it doesn't get that hormone signal. So again, good illustration of the importance of that one signal causing a really major difference in these kinds of plants. So auxin, one of the more important ones, better known hormones, also one that's got, as you might expect, some of the earliest data to support the role of auxin. So this is an experiment that goes all the way back to the time of Charles Darwin and his son, who were involved in doing experiments on these developing seedlings in the grass family, so maybe oats. And they would germinate these seeds and allow them, as you probably experienced if you're growing plants on a windowsill, plants are able to angle towards the sun. So this response growing towards the sun, this was an experiment to kind of investigate, all right, what is it about the plant that senses and responds to the direction of the sun and allows the plant to grow in that direction. So on the left here, this is an unmanipulated plant that starts off growing at an angle and then figures out that the light source is coming from the left and it angles to grow back in that direction. So it changes growth direction so that it grows towards the light. That's an obviously beneficial strategy if you're a seedling, is to head to where the light is abundant, and that's going to give you more resources through photosynthesis. So that's the normal wild type unmanipulated version. What else do you get? Well, if you cut the head off of that thing, so if you chop the growing tip of that, the signal is gone and it just grows kind of blindly upwards. So this plant will continue to grow even though it's missing the top. And the difference is that it doesn't grow toward the light anymore. So the result here, the answer is that something was important in the tip that is now missing. And because it's missing, that plant can't respond. So something was important in the tip. Moving on to the next one. All right, so we'll leave the tip intact, but cover it up. So this is an opaque top that prevents the cells in the tip from sensing where the light is. They're still intact, but they can't see the light. And in this one, again, ends up growing just straight upwards. So having light get to those cells is the important part, not just having the cells, but actually having them be the ones that perceive the light is important. And that's backed up by this other experiment here where the cap was more of a space helmet, where the cap can, or the tip can sense the light even though it's covered. So it would have the sensation of maybe contact from the cover or whatever it is, the different kind of atmosphere inside of that would be different. So it kind of replicated this situation, except now allowing the light to go through. And that showed them that that was good enough. So having the cells intact and perceiving the light was enough for them to angle towards the light. And then this one, kind of interestingly, was blocking out the cells along the stem. And they were actually not angling toward the light because they were covered, but these other ones were. So learning a lot about, not necessarily in this experiment discovering so much about auxin, but ties in with what we'll see later, that auxin is this hormone produced at the growing tip of plants. And so it's an important one that kind of puts the pieces together between sensing the light and growing towards the light. So auxins all together, this one, indole 1 acetic acid, is the kind of the baseline auxin IAA. And then these other ones going by the names of IBA, IPA, PAA. They're variants on the starting molecule with kind of similar effects in different plants. So auxin, relatively simple start. This is tryptophan. This is the amino acid tryptophan. And that's the starting point through a couple of these enzymes for making auxin. So rather short departure from an essential amino acid produced in the tips, the shoot tips, I should say, of plants. So typically auxin produced in the shoot tips and then transported away from that point. And some of the transporting molecules, we'll see some of these. PIN is a kind of transport protein that transports auxin, and it's transported in an asymmetrical fashion. So it's transported directionally away from where it was produced, and that gets the effect of kind of setting up a gradient where there's lots of auxin where it was made, and then it kind of diffuses out. It's transported, but also diffuses away from that location. So the plant can, in its cells, kind of tell how far each cell was away from that growing tip and get a different response. Just another illustration of that, showing a larger class of these families of pin proteins. So pin 1, 2, 3, 4, 7. Lots of molecules involved in transporting, and all these arrows, these directional transport of auxin in the root tip and also the shoot tip. It's complicated. We saw earlier about how leaves are initiated. So this is looking down on the leaf, or sorry, the root, shoot apical meristem, not root, shoot apical meristem, and initiating these leaves in the lighter color, the startup for new leaves. And that pattern is matched pretty well by the pattern of auxin distribution. So auxin is involved in signaling to cells that they're going to become leaves very early on in development. That's another one of its major functions. And we saw this before, this idea of apical dominance. So the tip of the shoot sort of communicating to the rest of the plant that the shoot is getting the primary growth, and areas near the shoot are kind of inhibited from growing. So if this shoot tip is growing here, the presence of that intact shoot tip prevents these axillary buds from growing out. And we saw that before. If you cut off the tip of the plant, then that removes that signal, and that allows these axillary shoots to grow out to the side. And this was an interesting paper that was kind of refuting a little bit of the general wisdom, the accepted idea that auxin was the main signal, basically coming from the tip and telling these side shoots not to grow. That was an important part of the signal, but it wasn't necessarily the whole thing. This study was illustrating that the presence of sugars, so just being the cells of the growing tip being a really big sink for using up those sugars, that was preventing a lot of this side growth from happening. So cutting the tip also prevented the sugars from reaching the tip, and instead they could grow out and enable these side shoots to grow out. So auxin definitely still important in this apical dominance, but the relative abundance of sugars also turned out to be pretty important. So auxin, as far as enabling cells to grow, one of its roles is that it loosens the cell wall. So in the presence of auxin, the cell wall can break down some of those side connections. So if you remember, we had hemicellulose and pectins that were holding these cellulose strands together. Those loosen up a bit and enable the cells to grow. So the cell wall, at least early in development, is in certain kinds of cells, is not strictly fixed as a rigid cell wall. It can actually become stiffer or looser depending on some of this chemical activity, and auxin can be important for changing that. Another role of auxin is involved in seeds and fruit ripening. So it's involved as shown here, or I suppose as not really shown here. The presence of seeds is an important signal for allowing fruits to develop. So we're talking about like fleshy fruits, and think about the contrast of how much waste it would be if the plant just developed a fleshy fruit without having any seeds inside. So basically having those seeds sets up a chemical signal, a hormone signal for later fruit development. So we have here a banana. On the right, this is a wild banana, not one that you'd ever find in a grocery store. Lots of seeds, not a lot of flesh in there. And this is better for the plant's perspective. So lots of opportunity for spreading seeds to start new plants, and just enough sugary enticement in there to get somebody to eat it and maybe spread the seeds around. What humans have done through cultivation and domestication is to take that ancestral form of the banana and turn it into one that's a lot of flesh and hardly any seeds. So the seeds in a banana are so minuscule you don't even notice them. They're these little black specks. They're not actually functional as seeds, and they're really not effective at giving the plant a signal for developing a flesh around that. So these forms of banana that we have, they're not really, not what the plant wanted to make ancestrally. They're one that humans have kind of tricked these plants into making, or figured out isolated plants that do this kind of lucky accident for us. So in the absence of this hormone signal, how do you get a giant fruit to be made? Well, turns out you can just take advantage of mutants. So this is an example from tomatoes. And on the right-hand side, we have this mutant that's not sensitive to auxin. So it sort of grows a nice fleshy tomato, whether it's got fertilized seeds inside or not. So there are no seeds in here. This is a seedless tomato, still developing at a nice size relative to the seeded tomato at the left. And this is the result of a mutation. So normally this tomato wouldn't grow big and red and juicy without having seeds inside. But because this one is a mutant, it doesn't know any better, and it goes ahead and grows big and red and fleshy, even without seeds. So same thing is going on in a lot of our seedless fruits. This is not something that natural selection would not really favor that over time. These plants making empty fruits full of nutritious, expensive sugars. But in the hands of humans and cultivation, we can take advantage of some of these mutants and get the thing that we're interested in. Another role for auxin is as one of our important classes of herbicides. So there's a chance you might have sprayed this on some plants in your neighborhood. One of the commercially available synthetic auxins is known as 2,4-D, 2,4-dichlorophenoxyacetic acid. Shown here, this is the 2,4-D molecule in colors, and then auxin over here. So it's a molecule that basically tricks the plants into thinking that they're getting auxin. And as a weed killer, it has an interesting function where it encourages the plant to grow, basically grow until it's out of resources and dies. So what you often get after you spray with one of these synthetic auxins is these plants are, they've kind of lost their control over the structure of their growth, and they tend to grow just out of control in kind of a spirally fashion. Just lots of growth and losing their single direction of growth and kind of bending over themselves. And in a short amount of time, they exhausted their resources and they die for that reason. There's a video I can link to about that. This is a plant in the process of dying as a result of the synthetic auxin application. Just kind of growing accidentally into a lump of wasted energy. So I mentioned there are a few different kinds of auxins. That one called IBA, down here on the label, indole 3-butyric acid, is another kind of auxin, and this is one that's often sold as a rooting hormone. So you can see again the hormone benefit here. This is a hormone that you can add to stems of plants and sort of convince them to start producing roots in a place where they wouldn't normally make roots. So you can convince stem cells to produce roots, and this hormone helps that out a whole bunch. So if you wanted to take a cutting from a woody plant and start it growing roots, this is your one of your big options here. All right, let me stop the video and we'll shift to a new one on new hormones.